Okay, I'd like to introduce Gabriel Kreimann in this, just like I was Tommy's student, so Gabriel was my student. At Caltech, was a brilliant student, helped this, and discovered these. In fact, one day he came to me with a cell that responded selectively, uniquely to 3 different images of then President Bill Clinton. So that later on turned into these Jennifer Aniston cells. Subsequently, he became a postdoc with Tommy at MIT, and now since 6 or 7 years is professor at Harvard in ophthalmology and neurology. Gabriel.
Thank you very much. So I'd like to continue with this theme of trying to build bridges between brains and minds and machines, and specifically I'd like to discuss the roles of recurrent and feedback computations in cortex. So in case I run out of time, here are the main players and people who have contributed to this work. I will highlight the work of 2 amazing grad students, Han-Lin Tang and Bill Lotter, as well as some of these other people here. At the heart of the main message that I'd like to convey today are 2 main claims. The first is perhaps pretty obvious, that over the course of many millions of years of evolution, our brains have produced circuits that do interesting things that we refer to as intelligence. And the second one, perhaps more controversial one, is that by studying and elucidating those neural circuits and biological codes, we can translate those biological insights into mathematics and computational algorithms to give rise to the next generation of intelligent machines. And I'd like to give a couple of examples on that. So this is a diagram that many of you may be familiar with that illustrates some of the basic connectivity in the primate brain. Christoph illustrated some of these ideas also in the rodent as well. And there's a very complex series of circuits here that are involved in visual recognition. And as both Tommy and Christoph have emphasized, there has been tremendous success in describing some of the basic aspects of computations that give rise to pattern recognition over the initial 150 milliseconds of processing. There's evidence at the behavioral, physiological, as well as computational level that we can capture many of these computations essentially in bottom-up architectures and feed-forward architectures that have then, also, of course, also given rise to the tremendous excitement in deep learning and deep convolutional networks. Now, we know very well that in cortex there are massive recurrent as well as back projections throughout cortex. And this is just one of many studies trying to quantify exactly how many connections we have in cortex that are recurrent, that is, that are basically at the same level of processing, or even feedback processing from higher visual areas back onto lower visual areas. So the question that I'd like to pose today involves trying to take some initial steps towards understanding why we have these connections and what they might be doing, and how, by understanding the biology of what these connections do, how we can illuminate the future aspects of computation in cortex. So in order to do that, I'm going to give 3 examples. I'll very briefly talk about the fundamental aspects of feedback computation onto primary visual cortex, then move on to visual search, and finally discuss in a little bit more length what happens and how recurrent and feedback computations can help in the context of pattern completion. Throughout, I'd like to bring essentially the 3 levels of analysis that David Marr and Tommy Poggio postulated, that is trying to understand all of these problems, both at the behavioral level, at the physiological level, as well as at the computational level. Why do we want to start with primary visual cortex? Well, as Christoph emphasized, and then Tommy also noted, a lot of the basic science that gave rise to deep convolutional networks originated by some of the initial findings by David Hubel and Thorsten Wiesel, studying the so-called simple and complex cells that in primary visual cortex. So I'm going to show very quickly an experiment that was conducted by Rick Bourne at Harvard Medical School, essentially a few steps away from the laboratory that David Hubel used to do his recordings, where he used a cooling technique to lower the temperature in area V2, V3, which is the main source of feedback onto V1, and then examine the properties of neurons in primary visual cortex sans feedback, in the absence of of this feedback signal. So this is just an illustration. You can lower the temperature of V2, V3. You can therefore silence those neurons, and then you can record the activity of V1 in awake behaving monkeys while in the absence of these feedback signals. So the first point I want to make is that a lot of the basic properties of computation in V1 remain unaltered in the absence of feedback. So if you look at the direction selectivity, if you look at orientation selectivity, if you look at the tuning width, some of this basic initial response is unaltered, consistent with the notion that, again, we can explain some of the fundamental computations in a purely feed-forward manner. However, when you start to look at what happens beyond the primary receptive field of the neuron, beyond the basic simple computations, you start to see quite notable and important differences. One of them is illustrated here and has to do with so-called areas around suppression. So if you start increasing the size of the stimulus, eventually the responses of the neurons become smaller. So that's illustrated in these area summation curves. On the y-axis, you have the firing rate. On the x-axis, you have the diameter of the stimulus. Larger is better at the beginning, and eventually larger is not longer better, and the responses decrease. It turns out that in the absence of feedback, if you don't have feedback signals, this inhibition that gives rise to this decrease in the response is significantly ameliorated. So feedback is playing a fundamental role in this constraining of the size of the receptive field and in the integration of signals beyond the classic receptive field of the neurons. These feedback effects are delayed with respect to the initial onset, so it takes time for signals to go all the way to V2 and then back onto V1. So we can use the latency of these signals as a signature for the timing involved in feedback processing. And it turns out that we did a series of computational exercises showing essentially where we can ascribe these feedback signals and trying to, as I said at the very beginning, try to read biological code and translate that into mathematical algorithms and computational ideas. So people have described these area summation curves as a ratio of two Gaussians. Essentially, one Gaussian curve describes the essential input. The other one describes the normalization operation, one of the fundamental operations in many of these deep convolutional networks. And it is in that normalization operation that we think feedback is playing a fundamental role. So by understanding the biology of these feedback signals, we can begin to write algorithms describing how feedback may be instantiating these inhibitory computations in cortex. So I want to move quickly to my second example now and change gears to talk about another fundamental problem where we think these feedback signals could play a fundamental role. And that has to do with the problem of visual search. Many of you are very familiar with this kind of Where's Waldo kind of games. This is a pretty difficult task. When you try to do this, it takes time. And this is one of the fundamental issues with clutter in visual recognition, whereby it's very hard to actually recognize objects in clutter, and you need to move your eyes and actually perform quite a lot of computations to reach for that. So inspired by a lot of physiological recordings in macaque monkeys, we postulated a computational model involving feedback to try to solve this problem of visual search. In the interest of time, I'm not going to be able to discuss the physiology in detail, but everything we're talking about here in terms of the computation was significantly inspired by the work of people like Bob Desimone at MIT, Manuel Trujillo, and many others who studied the role of feedback signals in macaque cortex during feature-based attention or during visual search. So very, very quickly, at the heart of this computational model, we have the traditional deep convolutional networks, in this particular case, the HMAX Tomipoggio kind of instantiation. And then what we do at the top is, let's say that we're searching for Waldo. We store what particular features are at the very top of the hierarchy are representing Waldo. And then when we do is inject feedback signals, illustrated here in the top right of the slide. We inject those very specific feedback signals, essentially pushing Waldo all over the image, pushing the particular features of Waldo all over in order to highlight those locations in the scene that are more Waldo-like and that will help us perform visual search. And there's a paper that's published on this if you're interested in getting to know more of the details. So here are a couple of examples where we're looking for particular objects, either in images that contain arrays of multiple objects and or in natural scenes. And what you're seeing there is the performance, or how many times you need to fixate, and how well the model can essentially find Waldo in these complex images, or how well the model can search for different complex objects by instantiating these feedback signals. We can do this either in the situation where we tell the model to stop looking, or the model itself has to realize when it actually found the object. We can do this in images that contain isolated objects, and to a lesser degree as well in complex images with natural backgrounds as well. And importantly, as I emphasized from the very beginning, we really want to connect the computational models with what's happening inside the brain, with ultimately behavior. So here's a psychophysics experiment trying to ask how well people can recognize and search for objects in complex images by measuring their eye movements. And we show here that this very simple computational model instantiating feedback connections can match human performance extremely well in this very simple visual search paradigm. I don't have much time to go into, there's a plethora of other algorithms and that people have put forward for visual search. Laurent Titi and Christoph Koch have done beautiful work with purely bottom-up saliency models, and a lot of other people have used different strategies for this target-based visual search. At the heart of many of these algorithms is the idea that you need feedback signals that are extremely specific to be able to search for the particular object that you're looking for. So these are two examples from the bottom up, the basic computations in B1, all the way to the highest echelons of processing. And I'd like to focus lastly on a third example of how we can try to connect behavior with physiology, with computational models. And that has to do with the problem of visual pattern completion. So I think pattern completion is at the very heart of intelligence. Here are just a few examples where people can complete patterns in the context of sequences of numbers, or the famous connected dots kind of game. We use pattern completion in social scenarios. From a second of interaction with a person, we can read them to a first degree reasonably well. We do pattern completion and extrapolation from partial information all the time. I'd like to focus today on the question of pattern completion in the context of visual processing. There's a number of different ways in which we can create images that are incomplete, and where, in order to be able to recognize them, we need to do pattern completion. And I'm going to argue, based on behavioral evidence, physiological evidence, as well as computational evidence, that we actually may need recurrent and or feedback connections to solve this problem. So let me start from the behavioral part. We created images such as the ones shown here for the toy school bus. This is using a technique that's common in the psychology literature called bubbles. Essentially, it's like looking at the world like this. So you have a few apertures through which you can look at objects. So you have a pretty severely degraded version of the object. And it turns out that our visual system, as you might imagine, is extremely robust to deleting huge chunks of the image. So here we're showing psychophysics performance on the y-axis as a function of the amount of occlusion. Each of those lines corresponds to different exposure times, how long we present the images for. And you can see that you can go up to 80 or 90% occlusion. So you only have 10 to 20% of the information in the image, and we can still recognize those images quite well. In this particular case, there are 25 images. There are 5 different categories. People are doing a categorization task, and people can do extremely well despite very, very heavy occlusion. So I'd like to now very quickly show you what happens inside a human brain while people are performing this type of pattern completion analysis. And Christoph already alluded very briefly to this notion that in some particular cases we can interrogate the human brain invasively by working with patients that have epilepsy and who have electrodes implanted inside their brain in order to monitor or cure them from their seizure events. So this gives us a rather unique opportunity to record either the activity of single neurons, or in this case, field potential signals invasively from different parts of visual cortex. So here's one example first, without occlusion, just what happens when you present pictures, what's happening inside the human brain when you see objects. So each line in this plot corresponds to one presentation of one particular picture. There are several repetitions. There are 25 different pictures in total. The color map over there represents voltage. It indicates how active this particular electrode was for the aficionados. This electrode is in the inferior temporal gyrus, an area that we know is fundamental for object recognition because lesions in this area render a significant impairment in object recognition capabilities. If you average all of these trials and all of these different pictures within each category, you get a signal that looks like this. This is the intracranial field potential as a function of time. So we get a very selective and strong signal that happens very fast, consistent with the feed-forward models, consistent also with the behavioral experiments. So we have a very strong selectivity, invariance that I'm not going to show here, and a very rapid response. So what happens in the human brain when we significantly occlude the objects and we present only partial information? So again, here's just one example of one picture, 30 plus repetitions of that particular picture showing the potential as a function of time from another electrode. So here I'm going to show you a few single trial examples of what happens when you only present about 10% of the image. It turns out that to a first approximation, although there's some variability from one picture to another, the signals remain largely the same, meaning that the signals in these highest echelons of visual cortex remain strongly selective despite these very strong degrees of occlusion. If you pay more attention to the details of the waveforms, it turns out that there are some important differences. Most notably, there's a delay in the signal. So those numbers there represent the maximum voltage, and it turns out that that is significantly delayed with respect to the responses to whole objects. Remember that I had shown you at the very beginning that these delays are consistent with the role of feedback signals. So I just showed you a couple of examples just to convince you. Here are all the trials with whole objects in the same format as before. Here are all the trials with occluded presentation. There's variability from one trial to another because the images are actually different. The positions of those bubbles are actually different. But consistently, there is a significant delay of about 50 milliseconds or so involved in the essential physiological signals underlying recognition of occluded objects. So this is more data, multiple electrodes. I'm going to skip these and go very quickly through these. We consistently see these delays, which we interpret to imply that we need additional computations, perhaps these recurrent and or feedback computations. So in the human brain, we cannot cool an area. We cannot inject electrodes and lower the temperature of an area like Rick Bourne did in macaque monkeys. It's illegal to do that. Our alternative here is to use a technique called backward masking. Essentially, you present a picture, and very briefly after that picture, you present a mask. And it turns out that this has been hypothesized to essentially interrupt feedback processing. So at the behavioral level, we repeat the same behavioral experiments, and we conjecture that if feedback is important to recognize partially occluded objects, we would be able to impair performance significantly in recognition of partially occluded objects when we perform this experiment under backward masking. And indeed, the upper plots here correspond to the psychophysics data, behavioral data, in the absence of masking. And if you look at the lower right version of this figure, on the y-axis you have performance, on the x-axis you have the interval between the presentation of the picture and the presentation of the mask. And if you present the mask at the right time, essentially to be able to impair feedback, we see that performance is severely degraded. In other words, if you inhibit these putative feedback signals by presenting a rapid mask after the stimulus, you cannot complete objects, you cannot do pattern completion, you cannot recognize these objects well when they are heavily occluded, whereas you can still recognize objects pretty well when they're not occluded at all. So just very quickly, I want to point out that there's a connection between what's happening inside the brain and what's happening at the behavioral level. So we can go back and take one of those electrodes that showed a very strong selective signal and ask for each picture how much of a delay there was in the physiological signal. Remember that I postulated that that delay in the physiological signal may be correlated with the need for additional computations. At the same time, for each picture, we can measure behavioral performance and what's the effect of backward masking, to what extent backward masking impaired recognition performance. Bear in mind that we do this across different subjects. So we have physiology data in one group of subjects, and we did this psychophysics experiment in another set of subjects. So despite these limitations and these differences, it turns out that there's a weak but significant correlation between these two worlds, what's happening inside the brain and what's happening at the behavioral level. Essentially, when an image was when a particular object was more impaired at the behavioral level by masking, it was also more delayed at the physiological level. And that's what these scatter plots are showing here. So let me now come back to computation and argue that purely feed-forward models also struggle with very, very heavy amounts of occlusion. This is the behavioral data that I showed you already, performance as a function of the amount of occlusion. This is how well you can recognize the same set of objects using just the pixels. This is how well you can recognize them with the HMAX model. Bear in mind that the HMAX model has 0 free parameters. We're not trying to train these parameters at all. These are, for the aficionados, these are different versions of the so-called AlexNet deep convolutional network. This is the Pool 5 level or the FC7 level. These computational models start to perform better, but they still fall significantly short of human performance. So they still significantly underperform humans in recognition, particularly for heavily occluded objects. So at the computational level, we'd argue that we need something beyond these purely feed-forward architectures for recognition. So just to try to visualize that, here's a multidimensional scaling 2D rendering of the representation of one of the set of pictures that we had in our experiments from the top level of the AlexNet network. So here are all the different whole objects, and you can see that they're pretty well separated in this space. So it's pretty easy to distinguish all of these different pictures when they are whole pictures with this highest level of AlexNet. In contrast, when we look at the representation when they are occluded, it turns out that they cluster essentially in the middle, and this is what gives essentially to a very poor performance in recognition of these heavily occluded objects. Again, it turns out that we can measure for each picture how difficult it is for the models to recognize them, essentially how distinct is each picture from the category mean. Again, there's a weak but significant correlation between what the model is saying, between how difficult that particular occlusion problem is for the model and how much delay there is at the physiological level. How can we rescue performance, given that at the behavioral level and at the physiological level, I argue that we need recurrent computations? How can we use these ideas about recurrent computations to rescue and improve performance for recognizing heavily occluded objects and perform pattern completion? It turns out that there's a very nice and old literature on Hopfield networks. These are networks that are interconnected in an all-to-all fashion and can perform pattern completion extremely well. So taking inspiration from these ideas as well as from the behavior and the physiology, what Bitt-Lotter did essentially was take the upper level of AlexNet and add recurrent computations at that level. So he added all-to-all connectivity and retrained the network to learn those weights in order to try to recognize heavily occluded objects. So I'm going to go through this very quickly in the interest of time. Here's the pictorial rendering that I showed before for all of these different categories. And this is what happens in this network over time, because now with that we have recurrency. The network changes its state over time. And you can see that over time there is better and better separation of these heavily occluded objects, which suggests that we may be able to recognize them at the computational level. And indeed, when we go back and plot performance as a function of the amount of occlusion, it turns out that this adding this recurrency step and adding these recurrent computations onto the upper level significantly improves performance and also matches human performance. Not only in terms of overall performance, this performance improves over time over these multiple recurrent computations, and also at the level of correlating performance between behavior and the computational model for each single individual picture. So I'm going to stop there. I just want to summarize the 3 main points that I wanted to make. Here are 3 examples about ways in which we can try to read out biological codes and translate those biological codes into computational codes. The first one had to do with the very simple basic computations in primary visual cortex, the sort of ideas that have inspired generations of deep hierarchical architectures, and we have taken initial steps to understand how feedback can help and act in primary visual cortex. The second one was by showing how feedback can help in the context of visual search with cluttered scenes. And the third one is in the context of pattern completion and how adding recurrent computations onto the computational models can help us understand and perform better in heavily occluded object recognition tasks. Taking a step back, the main message that I'd like to convey is a rather positivistic one, arguing that ultimately by understanding brains, by understanding neuroscience, we will be able to design better algorithms. We will be able to help alleviate the tremendous human disorders that afflict the human brain, and ultimately perhaps understand better who we are. Admittedly, I put many, many years there. We're not quite there yet. It will take us a very long time to really translate biology into computational algorithms. But I think this is not only a plausible path to go, but actually a necessary one. And finally, I'd like to show the pictures again of the main people who have done the work. Thomas Micone here was involved in the visual search experiments. I mentioned Rick Bourne and Joe Nasi and Camille Gomez, who did the work on primary visual cortex. And Bill Lotter and Han-Lin Tang were the main people involved in all the work, physiology, behavior, and computational models that have to do with pattern completion. So I'm going to stop there.
A lot, Gabriel. We have time for questions, so please raise your hand.
So why add connections only among the top layer neurons and not going down to the lower layers? That's what I would have expected.
I assume you're referring to the last part in the computational model.
Yeah.
Indeed, this was our starting point to, as a proof of principle, show that adding any kind of recurrent computation and training those recurrent connections can help. Ultimately, as you point out, there are recurrent connections and feedback connections everywhere, and we still have a very long way to go to understand those. But we'd definitely like to do that, and we'll invite everyone to think about what are the computational roles of feedback connections throughout, which we know are fundamental and ubiquitous in biology.
This works? Yeah. Thank you for the nice talk. So I was wondering that you showed, it's very interesting that in spite of having occlusions, the activity of neurons remains the same. So the occlusions can be similar to extrinsic noise. And a lot of computational models have shown that when you have feedback connections, you can have internally generated noise like chaotic dynamics. And do you see that such internal noise plays a role in computation? Or pattern completion, for the matter?
Yes. Yeah, it's a good point. It's a good question. I'd be happy to discuss further. We haven't really thought about occlusion as noise exactly, but I do like the idea of thinking about it. So I would say that at the physiological level, what we're probably looking at is the end point, the result of that computation, and the fact that at this very last stage of visual processing, these signals are quite robust to what you call noise, to this heavy amount of occlusion. At the computational level, that noise or that variability does significantly impair performance. Things are really clustered at the beginning. Things are really obscure at the beginning. And what we think is, and sort of the mathematics of this is laid out in the Hopfield networks, is that by adding these recurrent computations, the system learns to separate these patterns and bring the representation closer to a more robust representation that's similar to that for unoccluded objects. Yes.
My name is Jürgen Schmidhuber from the Swiss AI Lab. Of course, there are many engineering approaches which also use recurrent networks to better understand images, to selectively direct attention to certain parts of the image such that it can be better explained, such that it can be better recognized. And these are driven by pure engineering principles and mathematics. You have a goal, you want to find your object, you have selective attention, which means some sort of recurrent network. And these guys are not at all inspired much by neuroscience. How will you see the dichotomy that we have there? So on the one side, the pure engineering guys who just solve their problem, realizing that they need recurrent connections for solving it better, and then the neuroscience guys who also realize the importance of recurrent connections, but there is not actually a lot of interaction between these groups.
Absolutely. I think it's fantastic that I think there will be plenty of ad hoc solutions coming up from engineering that will work. There are amazing programs, much to my, this is something I don't like, but I have to admit there are fantastic computational programs that play chess much better than most of us, and we've learned nothing about how humans play chess, despite the fact that the computational algorithms do extremely well. So sure, absolutely, engineering can find out a lot of solutions. Some of those may or may not overlap biology. Hopefully we'll learn from engineers and will inspire what kind of solutions we might look for. Hopefully some of the biology will help inspire engineers as well. And there will be solutions that have nothing to do with each other, and I think that's perfectly fine. I think ultimately for some of the very challenging aspects of intelligence, we have a system that works. We have a system that solves all of those problems, that is our brains. So I think that if we can sort of dig in and find out how it works, we should be able to translate that into computational algorithms, and we should be able to help and accelerate engineering so that they don't have to spend decades and centuries sort of trying out different ad hoc solutions, which can actually look at how it's actually implemented. That said, if there are solutions that have nothing to do with biology and they work, that's fantastic. I'm happy to applaud those approaches as well.
Great. So let's thank Gabriel again.